after a month back with another video. Let's quickly get into it. Last time what we had done was acute myeloid leukemia updates of 2022. The first part, maximum AML update, I had covered in that. A small part of that was left, which I am covering right now. This deals with AML with recurrent genetic abnormalities. You all know, we have read in the previous lecture that a lot of emphasis has been paid to genetic abnormalities in leukemias and lymphomas, even in the Hem 5. So the group of leukemia that used to fall in mixed or biphenotypic leukemias has also been divided based on the type of genetic mutation that is present in that particular phenotype. So now we will not leave something as MPAL or BPAL or ambiguous lineage. We are going to define it after when we see that there is both B and myeloid and T expression, or myeloid and B expression in that particular leukemia, but it favors myeloid more. Then we are going to define it with a particular genetic abnormality and see whether that is present or not. Alright, so it is just not limited to finding two populations and leaving it at that now. So myeloid neoplasms associated with germline predisposition, a novel scalable model is introduced. So in all those AMLs where there's germline mutation as well as mixed phenotype, we are going to study about them right now, not AMLs with only germline mutation. So acute leukemias of ambiguous lineage, that is ALAL, and mixed phenotypes, MPAL, are grouped under one, now single category. Framework for molecular classification is laid by separating ALL slash MPAL with what slash MPAL doesn't mean there are two categories. What it means to say is it has clubbed MPAL with ambiguous lineage into one category and then defined it according to the genetic mutation present. So there can be an MPAL with ZNF384 rearrangement. This ZNF384 rearrangement can be seen in MPALs with B and myeloid lineage, or even in cases of BALL. So in leukemias where you get a picture of BALL as well as myeloid markers positive, ZNF384 is an important genetic mutation marker that is being studied. 50% of pediatric B or myeloid MPALs with fusion partners include TCF3, EP300, TAF15. These are some other mutations which are being seen in B MPAL, but the most important mutation being studied in MPAL with B lineage is ZNF384. Then there are ambiguous lineage with BCL11B rearrangements. These are heterogeneous and labeled as undifferentiated leukemias, and in wherever there is BCL11B mutation, mostly a T myeloid type of picture is seen. And it is also rearranged in 20 to 30% of ETP ALL. Alright. Acute leukemias with stem cell, myeloid or T features have BCL11B rearrangement. So ZNF384 is B myeloid, BCL11B is T myeloid. Other genomic findings are PHF6 mutation, PCAM mutation, 10 fusions are seen in MPAL. These are others. You might remember too, BCL11B in T myeloid and ZNF384 in B myeloid. MPO expression with intensity and pattern similar to that of early myeloid maturation is more strongly associated with myeloid lineage than uniform dim MPO. What they mean to say is a uniform dim MPO say better hai a strong intensity MPO in early myeloid lineage cells. 19, 22 and 10 strongly associated with B lineage. Assessment of MPO expression by cytochemistry or flow plays a key role in intersecting AML with minimal differentiation from T myeloid, MPAL and ETP. We all know about that. MPO is the strongest indicator to call something AML. 3% cutoff of MPO historically was seen in cytochemistry. Now they say that a 10% MPO cutoff will remove any ambiguity or any confusion. So they are advocating that a 10% cutoff can be made instead of a 3% cutoff. However, no consensus has been reached on this till now. So to summarize quickly, it's a very short lecture. Acute leukemias of mixed or ambiguous lineage are arranged in two families. ALL with defining genetic abnormalities and ambiguous lineage immunophenotypically defined. Novel genetic markers are listed for ALL, which I have shown. Lineage arrangement criteria for MPAL are refined to emphasize principles of intensity and pattern. So for this, we saw ZNF 384 and BCL 11B in BNT myeloid. Other genetic mutations also had mentioned. And as I told you, MPO, a strong intensity pattern in early myeloid lineage, is preferred over a diffuse dim MPO positivity called something AML. So to summarize, if you see, this distinction of genetic mutation initially was not present for ambiguous lineage or mixed phenotypic leukemias, is now being introduced in Heme 5. Initially, we just had a separate group of AML with recurrent genetic abnormalities. It is still there. We still have that, but now even for leukemias with which are suspected to be AML and are mixed phenotypic and have recurrent genetic abnormality. So even in them, we are seeing whether there is any particular genetic abnormality or not. So this is a very short topic. So I'll finish it over here and I'll come back with the next topic as soon as possible. Thank you.